to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 64th episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I have been in the bioanalytical LCMS space for over 27 years. I'm here with my co-host Dominic Guarino.
Hi, my name is Dominic Guarino, and I'm a Senior Scientific Advisor here at KCAS. I focus on ligand-binding assays and biomarkers and gene therapies.
Dom and I are Senior Scientific Advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS's services. I'm located in upstate New York, Dom is in Kansas, and our producer is in Missouri, so we are truly all over the map. We're thrilled to have you listening to the 64th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on the topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're thrilled to have you here, and we are looking forward to a fun episode today. To kick us off for podcast 64, Dom is going to go over today's podcast topic a bit before we jump into the news section. Dominic.
Yeah, we got a fun, jam-packed podcast today. We're going to look at drug approvals in 2022, so that's a little look back, and then we're going to go a little look forward and look at what Fierce 15 top biotechs that are going to be up-and-comers in the next few years. And we also got a little special treat to look back a little bit at that Fierce 15 from 10 years ago. So, John, jam-packed, and then we'll talk a little bit about how KCS is involved in all of this. But it's a great episode, and we're excited to get going. And, John, without further ado, let's just jump right into the news and notes. What's the latest on COVID?
Yeah, it's as usual, it's getting briefer each time, but it's still worth touching on. First up is next-gen vaccines. No more quick variant updates and RCTs may be required CBER leader rights. And this is really just a commentary from Peter Marks, who's the CBER director, talking about the future directions that the FDA sees for vaccine development. So they're thinking the emphasis is much less likely to be updated vaccines to address current variants and more longer term strategies to really look at improved vaccines to be prepared for future mutations, etc. And also the bigger part of it is that if they do go in that direction, the more of a return to randomized clinical trials to really assess the efficacy of any new vaccines coming through.
Yeah, I think it's just science.
Yeah, absolutely.
Yeah. I do agree with it's insane to do the same thing over and over again and expect a different result, right? So if we want to make broader, more effective vaccines, we have to do different things.
And they're out there. I mean, or people are working on things that are taking different approaches. I mean, there's a couple of nasal ones, nasal-based vaccines that are being looked at. I know there's other vaccines that are rather just going for the spike protein, are going for a more universal portion of the virus that then it's more variant proof, so to speak. So I mean, the work has been done out there, and I think it's BioNTech saying it's got, it's also looking at different approaches, and their next generation is combining the spike protein with a T-cell based vaccine to hopefully improve the response. So I think it's consistent with where the industry's going. I just think it's interesting that the FDA have basically said, here's what we're seeing, and we don't want these like quick releases of an update. It's much more important that we look at the longer term strategy for dealing with the virus.
Oh yeah, I mean, I think we've often just thought of it as a humoral response, antibodies, although you need, to some degree T cells or both T cells and B cell are involved in the humoral response. But can we get that true T cell immunity, memory immune cells and things like that? That's what we're kind of engineering, right? So it's to be determined, and I'm still optimistic that there's utility for all these types of vaccines. But to your point, we'd probably like to see the clinical trials maybe increase in size, or at least, let's not try to skyrocket, accelerate them if not needed.
Sure, yeah, and I don't think that's being suggested here. So
Yeah.
let's move on, because, like I say, the COVID stuff is it's around, but there's not a lot happening. The other
Well, you
piece
don't want to
is,
do a you don't want to do a little dance like Jimmy Fallon did? I thought that was kind of cool.
no, if it was after a certain time. Actually, I haven't been watching TV. My wife's out of town, so I've just been doing other stuff online and
Not that
reading.
I'm a Jimmy Fallon fan, but he did a funny dance, a funny parody. God bless him. I don't think you could pay me enough to do what he did, but maybe you could. But like you said, let's move on.
So the other COVID news is, we had, when we talked last time, we mentioned that the most recent Lilly antibody for treating COVID had its authorization removed, with Evishield still out there as a potential, but it's more of a prophylactic use of antibody, a more preventative thing. With the rise in different variants and the seeming lack of effectiveness of Evishield, its emergency authorization has now been revoked as well. I mean, it's no big surprise. They've sort of signalled that its efficacy was waning, and I thought it's interesting that the FDA said in instances like this is, don't can your antibodies completely, but just keep them available in case, as other variants arise, maybe it's effective against those in the future. Whether that happens or not, I don't know, but it does. It's one more thing taken out of our armory for dealing with the virus.
Well, it's a little surprising to me, but it was probably never sustainable to use a biologic across everybody. But let's hope someday it is, right, John? That would
Sure.
be
Yeah,
a good
yeah.
goal. And I'm still somewhat perplexed why more of these MABs don't work, or right, this
Yeah,
monoclonal
absolutely. That's
therapy.
the
But
weird thing to me too. I mean, we had a flurry of them, and they've all been rendered ineffective by mutation, but we haven't managed to catch up with that. It's interesting.
yeah, I mean, I think there's probably an evolution of the virus, but still, most of these aren't targeting the virus. They're looking for things that are causative, or preventing. So you do get infected, instead of having a whopping immune response, you dampen it so you don't get as sick, right? That's a lot of the mechanism here, right? They're
Yeah,
sopping up
yeah,
cytokine
yeah.
factors, but I certainly think it's I don't want anybody who's listening to think, oh my goodness, there's nothing out there.
No,
Should I get sick? Or even, there's still working on preventative measures, and there's still a battery of things that seem to work. And I think that's the hardest part here, John, right? Like, everybody's a little different. Every COVID infection is a little different. And then that makes it that much harder to do a regimen where we can't just cater to everybody yet.
yeah.
Right? We can't give you these 3 small molecules and these 2 large molecules, and me these 4 large molecules and 1 small molecule. But that might be what it needs to be like. So
Yeah,
someday,
and we
that leads nicely to our next. Yeah.
we know plenty of people are working on it, because a number of people are still talking to us about
Yeah.
applying their molecule or their biologic to COVID. So there's plenty of attention out there. It's just we're in one of those moments where the treatment options, if you're going into hospital, whatever, are limited. Anyway,
I'll
let's
read
move
my
on.
famous quote. It says, "If we knew what we were doing, it wouldn't be called research." That was Einstein, right?
Okay, yeah, yeah. That describes my entire life. I've been researching life for 60 years. So. So let's move on to the non-COVID. First up, and yes, this is obviously has impact on how the pharma research is done, and will likely impact what we do. But it's basically passage of FDA Modernization Act 2.0 clears the way to reduce animal testing. Now, there's been a lot of attention in the industry in terms of trying to minimize animal testing, looking at what models can possibly work ahead of going into humans, etc. And I know, particularly in the European bioanalysis community, there's a lot of talk in terms of, using as little as possible to get your answer. So basically, this bill, it doesn't ban animal testing, but it gives drug developers more leeway in using alternative methods to investigate the safety and effectiveness of a new drug. So I'm going to pass it to you, and then I'll chime in with more.
Yeah, I mean, I think it's just the future, right?
Oh,
I
absolutely.
can't, the small molecules. I think there are plenty of in vitro assays that can help you, test the safety of them. I think for biodistribution, there still probably has to be some animal work, but maybe it could be reduced, right? Just from
Hmm.
all around. And then, on the biologic side, it gets a little hairy, John. It's always been somewhat perplexing to me, um, to routinely use a rodent animal and then a larger animal, usually a non-human primate, and sometimes you get what you use the for instance, if you put a humanized protein into them, you get whopping amounts of antibody, and it really doesn't mean anything. And we make these highly sensitive assays, and we're already moving towards not having to qualify or not validate all three tiers of the
Sure.
ADA method, which I think is an important step. But this is progress, in my opinion, and
Hmm.
I'm not. We'll still have to do some, but I think there is a path forward to use multiple cell lines,
Yep.
in vitro stimulation of human PBMCs as well, and so there. And then we have to refine from maybe some of your traditional testing to maybe even more like a flow test or a PCR test, where normally it's just an LCMS method for PK, or some form of a ligand binding assay. So maybe there needs to be a little bit more on the testing side if we're going to just say, okay, let's not dose animals, because there still is a very important part to dosing an animal, right, John?
Oh, absolutely. Yeah.
Yeah,
I
still
think.
that, like, the toxicity piece of it, the
Yeah.
realistic, and then the SAD mat, right? The dose escalation and so forth. They still have an incredible amount of value to
Hmm.
understand your
Yes.
drug.
Yes.
So,
Exactly.
it's a step,
Hmm.
but I think we're still probably a generation or two away from being able to remove animals altogether. But I don't think any of us would be opposed to reducing the number of animals
No,
to support a drug
I
product.
don't believe so. I think the community's been in this place for quite a while now. I do. Yeah, I think it's a good thing. I mean,
Yeah,
it's. I mean, it also it will emphasize that what they say, the three R's: replacing, reducing, and refining the use
yeah,
of lab animals for faster and more accurate discoveries in drug development. That can only be good. I mean,
yeah.
you know, I mean, I know there's a lot of focus on unnecessary, suffering of animals and things like that, which is good. And minimizing uses is also, yeah. I mean,
And then there's another cool, right? They call organs on a chip. That's the
yeah, sure,
microphysiology
yeah,
system.
yeah.
See, but those are they're there, and they're getting better every day.
They're
But they're
coming.
still, yeah, they're not. No CRO. We think about trying to GLP validate that. That's going to make
That's
my head hurt,
well,
right?
that's exactly it. I mean, you have a mandate like this. The pharma companies will react a certain way, and then we, as bioanalytical people, as next step or for the several steps downstream, we will have to adapt how we then analyze what their particular test system is, or
Yeah.
test the drug in that, analyze the drug in that test system. So there will be challenges for us as a result of this, but I
Yeah.
mean, it's all good.
No, I think it is, and we're even still trying to figure out how to do true PK on some of the cell and gene therapies on the market. Because I say true PK because they can't. It is definitely a pharmacodynamic assay, and
Hmm.
a whole for the majority of them. But these, I've just really, this is, John, I've been following this a long time. Remember years ago they put an ear on a mouse, right?
Yep.
It was like, oh my, and not only that, like the mouse would respond, and maybe I hope I'm not freaking anybody out, but that is the next thing, is to get that on a chip and make it functioning. Why can't we do
Yep.
that? I do think
Yeah.
this. I think it's very promising, and it's really cool how much the field has advanced. And
Oh, exactly. Yeah.
maybe it's just we're old too. I don't know.
It's just another, here's the future coming. So let's move
Yeah.
on to the next thing. So this one, I think this is. I love this article.
Yes.
It's like, this is, if you have a male scientist in your family, this is probably a Christmas present for them next year. It's basically Withings Smart Scanner puts a urology lab in your toilet bowl, and I'm not trivializing it because it's, but this is the the thought process it prompted in me. It's to those who've always wanted to digitally monitor the bathroom health at home, you're in luck. Withings debuted a toilet bowl-based sensor that polls the biomarkers and metabolites released with daily urinations to provide a picture of hydration, nutrient levels, and wellness, in addition to ovulation, and cycle tracking. I mean, what man doesn't want to have something in the toilet bowl to aim at? And as a scientist, learn about what's in your pee. I mean, this is just like fantastic.
Yeah, it is. I couldn't agree more. I've always been fascinated by, maybe this is too much information, the color, the volume.
Oh, yeah. You
know anything, it's a sign of your health. It's very important to be looking at that stuff. So this is great.
So I am not trivializing it, but it's like it's got a microfluidic urine analysis lab equipped with a rotating cartridge-based system that forgoes the use of test strips and is cleaned with each flush. Described by Withings are on board with this. Described by Withings as easy as 1-2P. The rechargeable automated device is capable of running dozens of tests with modules that can be swapped out for different types. It's not actually approved in the US. I think this is actually in Europe. You can get it. Maybe next trip over, I'll be bringing myself.
It's like $500 or something.
Yeah, so it's also not cheap. That's the other thing.
Well, that's not too bad. I don't. Well, you say 500, but maybe it's like you got to replace that once a month. I don't know what that means, right? So.
Exactly. I think the starter kit is the 500. It must be the internal workings that you replace monthly.
What a fascinating article this is, though. Sorry.
Yeah, I mean, it does get you thinking. It's
and then there's others, right? Like on nutrients and dehydration. See, that's a really good one. Yeah. Go ahead.
I mean, it is good to monitor your health, and here's a fun way to do it.
I think I'm always like kind of on my son about drinking water, and he's really into working out a lot, and so I think this could even be good for. We worry about it too, where he's, we don't want him. You just worry about health in general, weight, right, and all that stuff. So maybe some of this would be even good for adolescents, not just aged men like you and I, John. Well, I absolutely think it would. I want one. Like, oh my goodness, I couldn't get it fast enough.
I mean, it's like when people get worried about whether he's dehydrated, it's like, go upstairs and pee and tell me how to IDs. Tell me how you're doing.
Yeah. Like, I'm like, your lips are so chapped, because that is a real hallmark, right? And so now I'd be able to give him a readout, and all he needs is a little more monitoring now. But anyhow, I think this is how we get better global health.
And last thing is, and it's obviously they've put a lot of thought into it, and that it can recognize different users and things like that. So it's I don't know how it does it, but it does say there. It has UScan can distinguish between various user IDs thanks to its Stream ID feature. Low energy radar sensors embedded within the reader measure multiple variables to identify an individual's urine stream signature by detecting movement and distance of the stream. So anyway, it's
John, it's fascinating. If I do get one of these, I'm going to make sure no outsider uses my toilet with my wife and I. But I think it's great. Yeah, we can move on. But it's like my mind's racing. That could be circulating tumor cell tests over time. Think about it. You can
absolutely. Yeah. So anything that you excrete in urine, I mean, you can be detecting, and that could be really useful. Yeah, I mean, it's like you say, you have there's the entertaining side of it, but the actual value of it, it could be really, really.
It's got to be good for diabetics too, I imagine.
Exactly.
Yes. And then it's got a little centrifuge in it. John, I am going to have to research this a little bit.
Yes. I missed the centrifuge piece, but yeah, it's yes. I told my wife I'd found this, and she's like, I'm like, I've got the best article ever to discuss in the podcast. I can't wait.
Wait till you see your stocking next year.
Oh God! And if I don't get to next year, I'm going to be sitting in the corner so grumpy. It's like, where's my UScan? I asked.
We're having too much fun. I just think my wife would be like looking at me like, "Are you for real right now?" I'm like, "Honey, I care about you." We got clever experiments is our next article, right?
Yes. So this is more in your realm. In clever experiments, scientists use gene editing to treat heart disease in mice. So, I mean, we've got gene editing in various diseases, but in a recent publication in Science, Eric Olson, Simon Lebeck, and colleagues from UT Southwestern modified a gene in mice that encodes an enzyme that, when chronically overactive, can lead to an array of heart diseases. The enzyme known as CAMK2 delta regulates heart function as one form of CAMK2, which plays important regulatory roles in many organs of the body. This was one where they didn't want to turn the gene off completely, so they did some substitution within the DNA, so altering the code for 2 points in the enzyme that stopped unwanted oxidation and the protein's overactivation from happening. And so they did a study for mice with heart injury, and showed that the mice who got that editing treatment recovered faster and showed fewer signs of heart failure compared to those who didn't get the treatment. So their point was, this is a proof of concept thing. It's not a treatment that's ready to go all the way through development, etc. But it's another, like, say, clever thing that shows what the potential of what gene editing can possibly do in the long term for us.
Yeah, it's they don't talk too much about the exact mechanism, but I imagine it's some sort of CRISPR-like or something, right? But gene editing in general is like, 10 years ago, I would have said, keep that away from me. There's no way you're putting that into me. And now it's kind of like, man, it's working, right? And they even touch on it a little bit. And part of it is they're now realizing, like, delivery of the gene is not a problem for immuno-oncology and other solid tumors and things, but when you're the heart, it's the opposite here. And so editing is the problem in the solid tumors and things like that, but not so much in the heart. And that makes sense, right? One's a much more the heart is probably easier to get. Let me rephrase that. It's not easier to deliver it. It's just more. I think there's more risk, right? Because of and really they talk about looking at scans of the heart, make sure it's not enlarging. There's no physical, and 260 days. So it's really, this is some interest. This is how we go from isolating monoclonal antibodies to large molecules being. We'll talk about that a little bit. I think it's almost half or 40% of the approvals to cell and gene therapies. To these are the early stages that pave the way for true gene editing, and it's a who's who. It's MIT's involved. If you don't know it, the Southwestern Texas or Texas Medical Center. They're an incredible network. Not to be, nothing against MIT and the Broad Institute. They're obviously probably our best institute, but if you don't know what's going on down in Texas, they have an unbelievable amount of research at not just UT Southwestern, where one of the colleagues is from, but all of Texas is a mecca for it. So I digress a little bit there, but this is the next wave. This is how we get from, it's still probably a little bit away, but maybe my grandchildren are able to have some effective gene editing therapeutics. Certainly, I hope I'm around to learn about it, right?
So for them, it's like really trying to find, like you say, it's not so much the editing is the challenge here, but the actual delivery. So viral vectors are not the way to go. So they need to find a more non-viral approach to get.
You can't have your heart swelling, right? Like a tumor could potentially swell to X amount, and there's some pain and all that, but your heart starts to swell. That's death. I believe it's similar to what happens with pneumonia, but anyhow.
Anyway, so
this is why they don't let me in medical schools.
So it's just another thing in the future, and then this is the final. Oh, there's one more, I suppose. Last non-COVID is cancer deaths. Cancer death rate dropped by a third since 1991. A study showed. This really is a reminder of why we're doing what we're doing. I mean, so much of, I have to say, as a bioanalytical person who's focused on and loves running samples, etc., cancer studies are a pain in the neck because the samples always trickle in. You have 10 at a time or 50 at a time, and then the assay goes down. You have to bring it up a month later for another 10 samples. But it's vital, and this is why it's important. And so the recent it was the American Cancer Society looked at rates of cancer cases and deaths in the US, and they basically their conclusion is that because of the way treatment is advanced in oncology, there's been a 33% decline in the cancer death rate since 1991, saving an estimated 3.8 million people. I mean, that really helps bring home the importance of what we do.
Yeah. It's like 10% of our population, right? So
Yeah.
that's unbelievable.
One
Wow.
is it 1%?
No, we have
One.
380 million. Oh yeah, yeah,
Okay,
you're
yeah,
right. Sorry,
yeah,
my
but
mistake. My
it's
mistake.
still it's a significant number.
Yeah,
So
it's not
I'm not
small.
knocking that. So I mean, and obviously what we're seeing, and obviously the main topic we'll touch on that. There's so much. I mean, medicine has progressed a lot since we got involved 20, 30 years ago, but there's so many more different approaches coming through. This will only improve. The numbers
Yeah,
will only get better, is what
I
I'm
mean,
trying to
it's
say.
no longer a death sentence when you get
Yeah,
cancer, right? This stats are
exactly.
showing you got like a 1 in 3 chance, which is a heck of a lot better than it was in 1991, right?
Yeah, I actually bumped into one of my ex colleagues, and I'd seen him a couple of years ago, and he'd been diagnosed with, I think it was prostate cancer, and when it had metastasized, and he was given, um, no, so he said it was, sorry, I've got the timing all wrong, but he was given not very long to live, but it's actually through a combination of radiotherapy, surgery, and I assume chemo as well. It's in complete remission. He looked really good compared to
Uh,
how he'd looked the last time I saw him, because we worked together. Then obviously we both gone our different ways, and but we still live in the same village, and occasionally bump into each other at the supermarket. And I just saw him in the distance, and yeah, he looked so much better than the last time. So yeah, I mean, it's amazing the difference. What's out. It's amazing the difference that the many of the molecules and other species that we worked on, the difference they're making, and
yeah, nutrition I think is probably a
yeah,
part of it even, and
yeah,
then um, early detection.
yes,
I wonder
absolutely,
what
yeah,
it is. I wonder they give some stats, but we got to kind of move on. But I really wonder what it looks like. I imagine it's hard to track. Like, okay, what is the rate of leukemia, lymphoma? I think that's some of them have like non-Hodgkin's. I don't want to say it's been cured. That would be, but there's certainly, I mean, major
Mhm.
advancements there versus where things like lung cancer are still a struggle. Although I think that has come a long way as well. So, meaning, I wonder if they
Which is
stratify it. Yeah.
why when you talk about reading about new drugs coming through, a lot of them are going for non small cell lung cancer and solid tumor and all those. It's some of the easier to treat
Yeah,
ones.
well,
Like
I
there's
think the
different
meds, yeah,
genetic code.
the meds are keeping people from dying of heart attacks. So what you got to
Yeah,
die of
yeah,
something. But that's
and then
now I'm getting a little morbid. And speaking, I think let's touch on this last one because I think it's important to just be real about what's happening in our
Exactly. Again,
environment. Yeah,
this has been so. Well, let's move on. I think the oncology thing is definitely is. It's here's why we do what we do, and here's the value it brings. But the industry has had a rocky last year, and I think Fierce Biotech, which is one of the newsletters we follow a lot, started a layoff tracker, and they basically said so. We've gone from like the in 2021 there was so much happening in biotech. There was money, there was I in initial public offerings, so the industry was awash with cash, but it's almost like it's not come full circle, but obviously things have moved on a bit, such that 2022 number of the as 119 companies
jeez.
actually laid off people in 2022 in terms of biotechs, and it's not just small biotechs as I'll touch on very briefly, but some of it there's a mix of things. So it's like investments being withheld, but a lot of it has been readouts of drugs that haven't succeeded. So where companies have basically been their natural cycle, they've raised money, they've got drugs into phase 1, phase 2, whatever, and then they've shown that actually their theory behind that drug hasn't panned out. So that's been a feature of it too. We've seen where you get, well, partnerships are a key part of pharma research, where a small company will stumble on it. They'll have a theory, they'll advance molecules. Large pharma like the look of those molecules, they'll partner, but if things don't pan out as they predict, then quite often you'll see those partnerships dropped, and there's been a number of layoffs as a result of that, because obviously the money stream that the smaller company was relying on is gone. So there's been a number of contributory factors, but it doesn't take away from the bottom line that 119 companies have laid off people through 2022, and it's still continuing into January. So just this week, the first half of this week, that we saw layoffs from Atomwise, Finch, and when I say it's not just small biotechs, Merck KGaA being the Merck based in Germany, they cut back in their research site in the US. So it's something we'll keep watching on, because obviously it has an impact on us as a company, and you have go through cycles, and this is just one of the cycles we're going through at the moment.
Well, a couple things. One of the benefits of being in a CRO is that when layoffs happen, there still needs to be productivity, and so our business may see an uptick due to some of this.
Yeah, sure.
But the downside is working in a CRO, we're 24/7, 365. Let's be clear about that. I think that's the trade-off, right, John? There's no sacrifice. You have to have some sacrifice to have that sort of stability, if you think of it that way. What got me thinking is you said something very powerful. It's, hey, drugs do fail. So maybe this record, because it is, I think, a record number, maybe this is more a cause of, hey, for 2 years during COVID, no drugs got killed, right? So all of that research is now being performed, and guess what? Now it's just maybe leveling out. So I don't know if this shouldn't be a gloom and doom panic moment, I don't think. I think this is more just for a couple years, no drugs got we're not able to research them properly, or they just slowed down through clinical trials, right? The whole machinery kind of came to a grinding halt as we worked through COVID, and a lot of the clinical trial work was focused on the vaccine world.
Yeah, I think there was slowdown. I mean, there were some failures, but it wasn't as many as you tend to read.
Well, that's what I mean. Like, so now it's just, we see these numbers, but I'd like to see, okay, what was 19's and then 2021, where it probably dips incredibly low for layoffs, right? And then we're just kind of coming back out. So I don't see it as now JP Morgan meeting in this month did show there were some mergers and acquisitions, but it was low, and the outcome was it could be a tumultuous 2022, but I think we're 2023. I misspoke there, but I think like we're oftentimes the pharmaceutical, biopharmaceutical, they just flow with how the general economy is going. So
and I do remember that when we started to see layoffs, etc., partway through 2022, and investment declining, there was definitely a couple of comments about it's like almost like the industry's got too big in the terms of the number of biotechs, and this is a righting moment. So that's probably part of it too. So there's a number of factors. It's just interesting to monitor how the industry is doing, and this is, like, say, we've gone from the contrast of 2021 to 2022, and 2023, like you say, could be tumultuous. We'll just watch it and help where we can. And
I actually, as the sentiment is, it's not like there's a lack of funding. It's just that people are starting to get it. Not that no one was spending superfluously. It's just people are getting a little more conservative, and that tends to.
I'd agree.
But remember, the difference between a bear and bull market is finite, in my opinion. So these are just things it's a natural cycle to some degree. So great, John, nice stuff. But what an exciting main topic we have. We ready to move on?
Yeah, absolutely. Yep, let's go ahead. One
of my favorites, John. We're going to take a little look back and then a little look forward, right? So we'll look back at what was approved in 2022, and then we're going to look at Fierce 15's who to watch and I think it's 2022. We couldn't have the Fierce 15 in 2023. So
Well, it's they announced at the end of the year.
yeah.
So yeah,
So
2022 really
it's a
got into
little, and then we're going to actually, we got a little surprise up our sleeve to look at some where are they now type moments. But
yeah.
John,
Yeah.
let's dive into. You did so much work on this. I'm going to let you. I don't want to steal your thunder, but I
No,
even
no,
wrote some other notes and stuff. But
it's not.
I'd like you to go through the list because it's always really, it's really cool to see. We had 42 approvals, I guess, right? And so
Well, it's really interesting, isn't it? It's like you can't even get agreement on the number of approvals,
yeah,
and
yeah. Maybe it was
I
45. I can't tell.
I pulled the list from in previous years. We've gone by the FDA site, and then because this was just a summary, and both endpoints and Fierce had the approvals in an easy to digest place. I actually went with Fierce, and Fierce listed 45 approvals. The FDA officially listed 37, but the differences are that the FDA doesn't list vaccines, um, gene therapies, or cell therapies. And there was also one very unusual approval, but I mean, again, it wasn't listed on the FDA website for ferring, where it was actually a way of treating Clostridium difficile. They were using it's like fecal microbiota. So,
Hmm.
it's
Saw
a
that one. I don't know if I want that one. No, go ahead. Sorry.
I'm not convinced either. But honestly,
Well,
again,
actually, the fecal treatments are great. If you have like, they say one of the, there's a side note. If they say one of the best treatments for like inflammatory bowel disease is that, but go on,
if,
John. Sorry.
yeah, honestly, if I get the infection and it works, then obviously I'll change my mind at that point. But that's one of the fundamental differences, and that's why I went with the Fierce, because it is important to highlight the cell and gene therapies and some of the things that's coming through. So of the 45, small molecule is still a big part of the industry. So 19 small molecule approvals. Looking at more conventional large molecules like proteins and MABs, there was 15 of those. There was also one mid-size. I don't know how to grade this. It's basically it's tirzepatide, which is the Eli Lilly GLP-1 analog, which we talked about 2 weeks ago, two podcasts ago, which the anti-diabetes with potential weight loss applications, where obviously you have semaglutide is the Novo Nordisk, tirzepatide is the Lilly drug, and where they've been where developed to treat diabetes, there may be a bigger market potential for them with the weight loss piece. So that's the mid-size. And then you've got one RNAi, two vaccines, one cell therapy, three gene therapy, and then two other, one being the microbiota. And there's a couple of like diagnostics or imaging type agents that the FDA includes, and Fierce did as well. Um, I was going to say something about the one of the vaccines was the Moderna, the other was the GSK,
Mhm.
measles, mumps, rubella, just to help increase the supply of measles, mumps, rubella. And then the gene therapies are interesting because with each one that was launched, they became the most expensive medicine, list price, in turn. So first one was a Bluebird Bio with a $2.8 million price tag. Then Bluebird, amazingly, had the second one approved at $3 million price tag, and then CSL and Unicure had a hemophilia B gene therapy with a 3.5 million price tag. But a lot of this is based on the fact that it's single
Mhm.
dose administration. That should be it. Anyway, I will stop rattling on. I'll let you have a.
I mean, I think I'm just taking it in. I'm gonna, crack down, drill in a little bit. Sorry, okay, drill out like and you're right. The price tag, it's hard to judge that. If is it, it seems high, but if it's a one-time treatment, I mean, that's just kind of how it works. But it's very powerful treatments, as you can imagine. But one thing that caught 19 small molecules, John. If we're going to look at
Yep.
let's
Yep.
look at the Fierce numbers because I always like to make it larger. 36 sounds so low relative to 45, but the 19 small molecules still dominates, John. Right? I
Yeah.
mean, that's
Yep.
still there are 15 like protein MABs, But I think if people, there's a perception that that would be surprising. Don't you agree?
I remember sitting in company meetings years ago saying small molecules are dead. We need to focus entirely on large molecules, and I'm thinking, but that's not consistent with what I've seen when out looking at company websites and their pipelines and all this kind of stuff. It doesn't gel, and this still bears it out, and it's still a fairly wide range of
Yeah.
applications. I mean, you got insomnia, dermatitis,
Yeah. Psoriasis,
you
right?
know,
Like,
like and
yeah.
anemia. There's I
Obviously,
can't
cancer,
remember.
lots of cancers.
There's rare disease. There's
Yeast
oncology.
infections, right? Yeast
There's a really interesting one
infections.
that I don't know if you saw the Novartis one, which is lutetium viplovatide. I can't. I'm not. It's print's really small. I can't read the full name, but it's interesting. It's a radionuclide embedded in a small molecule, but we've seen that there's a number of companies looking at radionuclides attached to antibodies, almost like an ADC approach.
Yeah.
This is a small molecule, but so there's like I say, the small molecule realm is still really interesting in terms of what people are doing.
Yeah, and so, like I said, it's from mental health to cardiac to,
Yep,
any sort of allergy, immunology, immuno-oncology.
lots of oncology. Yeah, yeah,
It's pretty cool. And then I think what strikes me as, like, there's an ADC on here, right? And that's I think if we were trying to predict what this would look like in 10 years, I think you'd see a lot of ADCs on here,
sure. Yeah,
which is just great. That's a win for both of us, I guess. But and
and
maybe not that's an antibody drug conjugate. I hope everybody in RS knows that. That's a combination of a small molecule payload that's attached to some sort of protein, typically a monoclonal antibody, that would then bind to the tumor and then deposit the chemotherapeutic just locally versus systemic. So real
yeah.
powerful stuff, right?
Even that we're seeing lots of different approaches with the ADC.
Yeah.
So what was a fairly narrow umbrella in terms of how people were doing it. There's lots of it's evolving quite
You,
rapidly,
you
the
know,
ADC field.
yeah. What part of before we got on the air here, I was a little giddy because it's been a weird week. But part of my week was there's a fascinating amount of drugs that are now in like a prodrug form, right, John?
Yeah,
We
yeah,
had some
yeah,
great things where one small molecule is given to activate a small molecule payload at the site of the tumor, which is just fantastic stuff. Or even like um, antibodies that have inability to bind until they're in the right tumor microenvironment. So they basically will stay in circulation. You can keep them systemically much longer because you add a peg or something to them. And then they don't bind until they're in the right microenvironment to cleave off what is essentially like a cap on
yeah,
the
yeah, yeah,
antibody, and then it binds. I mean, this is awesome. This is where we're bigger, badder, better, right? Like every drug that might have failed over the last 50 years now has the ability to be kind of reanalyzed. So I digress a little bit, but it's really exciting to see so many small molecules, so many large molecules, and then we haven't even got into like where I probably dedicated most of my life, right? That's the gene therapy pieces, the gene therapies, which have, there were. The vaccine world had a couple, but one mRNA, as you mentioned, one cell therapy, and three gene therapies.
yeah,
The gene
yeah.
therapies are awesome. I kind of looked into them a little bit, did a little more research on those. Um, you mentioned the two Bluebird. Man, are they really cool stuff? They use autologous 34 cells, so hematopoietic stem cells that they use an LVV, so a lentivirus system, and it's just amazing what they do. And it's a one-time therapy, and it engrafts back into the patient, and it's being used in some real rare diseases and so forth. But that therapy, John, over time, it probably takes a month to generate the cells. We're going to do that in 14 days, 7 days, and the
Hmm.
price tag will go down from three
Sure.
million to a
Yeah,
more reasonable price.
yeah,
But that's great. And then the other one was the Hemogenics. There is an AAV. It's a gain of function for liver proteins. They don't. It was really hard to find. I researched for a long time for me, which is more than 5 minutes, right? Spent quite a bit of time trying to figure out how this works. You can't find. I could not find it. I gave up. I think it's a lipid particle or a nanoparticle is how
okay.
they deliver it
And
now.
I actually missed one. There's a fourth gene therapy on here. I don't know
Yeah,
how I
it's
missed that.
ferring, right? I just see it now.
Yeah, sure, yeah.
And I don't know if you lumped it into maybe the cell therapy,
No,
but
no, no. It should have been gene therapy. I just
okay.
miscounted.
Because I did research that one as well. That's a non-replicating AAV-based gene therapy,
Yeah,
and that
yeah.
encodes interferon alpha 2b, which is a very potent treatment for a lot of tumors. In this case, they're using bladder or urotheliumia, I think I'm saying. But yeah, no, that I looked into that one a
Maybe,
little bit. That's
maybe,
a true
maybe,
AAV, though. Yeah, go ahead.
maybe I just missed that because it bucked the trend of going for the highest drug price ever.
Yeah,
So
no, that like they're the therapies that take advantage of the individual cells along with some sort of viral vector, they're going to be your most expensive, right? If it's
yeah,
just a cellular therapy, and there's still, I think one got approved there where they don't do any gene manipulation, but of course, all your CAR T cells and these
yeah.
HS, most of them, in order to really knock in the gene, and that's not the proper term, it's gain of function of a gene, or however you want to think of it. But I like to think of it as knocking in and knocking out, but that's somewhat dated, right? But anyhow, those are the combination of the two are where the price tag goes up. And of course, that doesn't mean we're not going to get better just traditional um, viral, some sort of viral vectors that are in encapsulated nanoparticles, and they're starting to make those a little more targeted. And also just, there's one mRNA on here, and we shouldn't, RNAi, we shouldn't leave those out because
No, no.
those are showing some great promise. But typically, you kind of got to inject them in the site of the infection.
I mean, that's one of the things that we're routinely hearing a lot more of. What's your experience on the RNAs and oligonucleotides and those kind of 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 entities? So
Yeah.
it's that's where things are
People are
trending.
putting RNAs onto
Hmm.
certain FABs, or even putting payloads on them now. I think you're just going to see a lot of cross pollination, and
Yeah, yeah.
it's just fascinating to see the evolution of this, and we get to not have to go through the blood, sweat, and tears of researching how they do that. People tend to come to us with one or two candidates that they've already put into an animal, and now they're looking to GLP validate those methods, and we get to pick it up, which is great, John, because we sometimes, I think you were on the call with me, we look at a presentation and it's like, wow, they just covered in like 5 minutes what probably took 20 years of research, right?
Yeah,
Or
yeah,
10 or
yeah,
something.
exactly. Yeah, yeah, yes, yes,
Yeah,
yes.
good stuff. I don't know. I
I
guess
think
we
that's
can.
something
You
we
got any more
probably should.
comments? Yeah, go ahead. I don't have much more. I
I
think
don't know
those
much
were
more. I
the
think
kind
let's
of.
move on to the Fierce
Yeah.
15, and I'll give a bit of background to that. So obviously, we look at Fierce and endpoints a lot because it's a central point where we can hear a lot of industry gossip and updates and things that it's a combination of we see news that impacts our customers that may impact us and just general trends that are again where we're going to be in the future. So they're both really helpful newsletters. Fierce have done their Fierce 15. I don't know how far it goes back, but basically they look at the companies are out there and identify 15 companies that they think will make a difference in the long term. And early in the year they did a 10-year update on their 2012 First 15, which we'll touch on. But it when you read through the 15 choices and the technologies or the thought processes that some of these companies are going through or what they're trying to do, it is amazing. I mean, it was to put some. It was. I always like to have the information to hand so I can talk about it, but to actually just go through that and carefully summarize what each company was doing was worth the time I spent on it because I learned a lot, not just in terms of who these companies are, but again, things that might be coming that might impact our bioanalytical teams in the long term. So it's really really useful. I don't know how we want to approach this.
Yeah. The problem here is, some of them we know, and so it becomes a little hard for me to want to comment on them because I'm excited to be like, oh, they're on here and they're on
Yeah,
here and
sure.
they're on here.
Yeah,
So we
yeah.
got to be careful. So maybe
Uh,
it's just maybe the best approach is just to go through the mix of work, right? Where these companies, it looks like there's still no surprise, a good share of small molecules on here, right, John?
yeah.
If my math is
I
right.
think let's work. We got the 15. Two of them I couldn't figure out actually
Yeah. Well,
because
yeah.
the websites say what they do, but ultimately it's not clear what they do from what they're administering. And
Yeah.
maybe
So
that's me.
the two
Maybe
you're
that's a
unsure of,
deficiency in me.
yeah,
But
Verge. Go
I'm
ahead.
not going to make any apologies for the fact that I couldn't quite figure out
No, no, because I
what was
don't
going
think
on.
these companies, some of them are still in like stealth mode, right?
Yeah,
That's the
yeah.
whole part.
So, and of the remaining 13, yes, there are one, two, three, four definite small molecules. So yeah, it's and that's really again
That
that's really
surprises us, right?
interesting
Like I thought they were dead.
because this is an industry group. I don't know how many people work at Fierce or whatever who've gone through what they know about all these companies and still decided that four of the most exciting companies within their 15 are doing small molecule work that people get dismissive about.
Yeah.
But when you read what they're doing, then it's some of the small molecule piece is really. I'm trying to think. It's really.
Engineered, right?
It's
So
yeah.
engineering comes to
This is
mind.
one of the trends I noticed. And sorry to jump on that, but it's like it's not so much the fact that they're producing small molecules; it's their upstream process to identify the small molecule that they think will be useful for a specific target. So it's increased use of genomic tools like CRISPR, medicinal chemistry, AI, and data crunching to really figure out the what molecule is going to impact the target they're after. So it's that mention of using AI etc. to help design the molecule was a big part of that.
Yeah,
And
no, I'm as baffled by you, not baffled, pleasantly surprised by the number of small molecules, and even one company here, this Amphista, they are bifunctional molecules. So it looks like it's small molecules combined. So
yeah,
even
that
though,
was
and then there's another gene. I think there's another ADC or some form of like delivery. So you can count them, but we can just quick recap. The number of gene therapies is not surprising, right? One, two, three, four, and then a cell therapy. I believe the Verge Genomics is a gene therapy,
may
as
well, it
is
may well be. I just
Aloxon.
couldn't figure it out
Yeah,
from
I mean,
the.
I see that, but I mean, you could really almost half of them are falling in a cell and gene therapy
Yeah,
space, and
yeah.
that wouldn't surprise us. It's just what is surprising is the lack of large molecules on here. I only see one true, large molecule company.
But if you
Uh,
look at that, that is, I mean, you're talking the generate biomedicines.
yeah,
It's
yeah,
basically creating de novo proteins to bind with specificity and drive the desired functioning. So basically, they're synthesizing proteins to go at their
yeah.
target. I mean,
But that am I that's there's no monoclonal antibody or humanized antibody companies on here. That's interesting.
yeah, absolutely. Yeah,
Well, maybe that's a nice segue into our surprise, right? It's the 2012, so 10 years ago. Fierce. Where are they now, right? So I don't know if unless we before I because you John thanks again for all the work you did on this. I mean, it's really cool to be just coming in and talking with you about this. So before we move off of it, you got any other
yeah.
comments
I
about it?
don't
But
think there's any
it's
other
a great
comment.
list. It's very
Aga-
cool.
Again, I think it's what's notable is the wide range where people are looking at. I think the Alzheimer's the shape therapies was an interesting one.
Mm.
I mean,
Hmm.
again they're looking at using machine learning to help with their gene therapy. It's interesting that I'm glad I added the category of who people are partnering with because
Hmm.
for these Fierce 15, um, really the
Every
bulk of
one of
them,
them, huh?
virtually every
Wow.
one of them. I think there's maybe one isn't, but virtually everyone is
At
partnered with
this
large
one.
pharma to help drive
Wow.
them. Yeah.
Wow. The lipid particle delivery system company, which of course is probably the cheapest to run.
Yes. Yeah. Oh yes. Oh yeah. That's a really Recode, which is again I didn't know
Yeah.
anything about them, but they have a different the nanolipid particle delivery is typically for lipids. They've got a proprietary
Yeah.
fifth one
Yeah.
that helps their helps deliver their therapies. So they'll I'm sure they'll partner with more people
Man,
as people
lipid nanoparticles for bioanalysis by ligand binding assays is a bear. So you can take
was
that, John.
and the nice thing is mass spec we can do them
I know, but
because the
it's
BD's team are
yeah.
the non GLP team are working more and more on the
I wouldn't even
nano
touch them. You can use a hybrid mass spec method or a mass spec method. But if
Mhm.
you came to me today and said, "Hey, we've got this ligand binding assay," I'm going to immediately try to convert you because they're just more. I don't know if accurate and precise is the right word. I think they're more robust on the
Yeah, yeah.
LCMS methods. But
So so
pain
yeah, that
pain me
that
to say that. Did you hear me have to take a deep breath? Like, oh man, no, I'm just kidding.
there's scope for both, but I'm always going to be in the side of LCMS.
Yeah, yeah.
Otherwise, I'm going to be saying I've wasted my life.
Well, John, I think my parting remarks on the 22 Fierce 15 is small molecules are far from dead.
Yeah, yes, yes. I really it's one of those things I will watch with interest where these companies end up, and obviously I hope that. I mean, I know we talked to some of them. I think less on the small molecule side, but hopefully we'll end up, collaborating with more of these, and you know,
Yeah, yeah.
and I do want to fight. I will let this go, but I do want to touch on that Amphista in Cambridge, UK, where you're right. The bifunctional molecule is
Mm.
two small molecules combined, so it's like one to target and then one to act,
Mm.
and they're already partnering with BMS and Merck KGA. So there's
That makes me say, oh no, why do we need the framework of the antibody
it
for
is
that?
yeah
Oh
yes
no.
exactly yeah, but I mean. But yeah, but ADCs work. I mean, so
No,
it's
I
just
think we're
another tool in the. It's like we said earlier, the approaches of that ADC type approach where it's something to target, to deliver a payload. There's a lot of different approaches, and this is just another one of them. So it's that's why I wanted to mention it because it really complements what we're already seeing a lot of emphasis on in terms of the projects that come our way.
Yeah, well, let's quickly go look 10
Sure.
years back, right? I
Yeah.
think this
Yeah.
is fun. We're going to go
Yeah.
back to the 2012 Fierce 15 list,
Oh,
and
and
the
I,
first thing I want to comment on, you can jump in, is just the number that I
first
closed.
of all, I'd like to say that I'm so grateful to Fierce for doing this stuff.
Yeah, it's we're geeking. It's and I'll thank you because John often does a lot of the prep work for us here, and I did more than usual, didn't I, John? But this is a lot of information you've got here, and I don't. Maybe I'll let you go, and then I can dissect it. You tell us
Go,
about the
go,
12, and then I'll share.
go ahead and what you. I mean,
Well, the
I
first
don't
thing
mind
I
it.
noticed is just how many of them are closed, truly
Yes, yes,
closed.
yes,
And one of them, the fact that there's no press updates or anything means it's closed, in my opinion. So there's
yes. Yeah. Oh,
one, two,
absolutely. Yeah,
three,
yeah.
four,
When it
five.
yeah when it says unclear and that really that that tells you that's just it goes back to what we were saying earlier about layoffs in 2022. This is just a natural
Yeah,
part of
yep.
biotech.
That was my
Things
point. Yeah, yeah.
companies will be established. They'll have a theory. They'll explore that theory, and it may have legs. They may partner, or they may run out of cash. I mean, there's a really interest. I one I really would like. So one of the ones of the closed is a company called Seaside Therapeutics. They were the last one on the list,
Hmm.
based in Cambridge, Massachusetts, looking at brain development disorders like autism and Fragile X syndrome. They closed in 2014, so only 2 years after the list was uh, was was
Full. Yeah.
announced. Yeah, whatever you want to call it. So one thing I want to make, I wanted to have locations just to know where people are, just because I like to have that stuff in my head. I went looking for Seaside Therapeutics because it wasn't clear. I then found that actually There's a company, there's Fragile X. Obviously it's a rare disease, but there's parents who are keeping each other up to date on what's going on with Fragile X. And there was like a newsletter saying, "Oh, Alos Therapeutics have picked up," and I can't remember the name of the drug. Sorry, I should have noted that. I picked up the name of the drug, and they're moving it forward. So I thought, "Oh, Alos Therapeutics, I'll check them out." It turns out that Alos Therapeutics are actually the same people who founded Seaside Therapeutics. So the drug's in phase 3 for what
I guess that, so you're right. Of the 5, one of them is technically still existing, right? Of closed, closed.
They just ran out of cash.
Yeah.
And,
so maybe it's, and that, I don't know, that seems not so bad, right? But then there's 1, 2, 3, 4, 5 essentially that got purchased or rebranded. The Myrogen to Viridian one, I can't tell if that's they say rebrand. So maybe it's really just 1, 2, 3, 4 of them. So we've got 5 closed, and 4 that are other companies now, or they just don't. That name's not along. They're part of J&J, Roche, and so forth. So that's 9 out of our 15 essentially. Which I don't know. I'm mixed about that. I don't think that's so bad.
Yeah, so Myrogen basically I think there's a merger with Viridian. They then changed the research emphasis. So where they were RNAi as Myrogen, now they're...
so really it's 10. I think we can say safely 10 of the 15 are technically either closed or they're just a completely different company.
Yeah, so and some of them, it was like their product was the large partner was so enthusiastic about their product that they bought them. Mm-hmm. So that was true of Aragon, bought by J&J in 2013. So it was really quick after the 2012 list.
Yeah.
But actually, you read more about it as it happened that J&J were interested in their main drug, which actually didn't pan out in the long term, and then they were spun out as Ceregon with the remainder of their portfolio. Others are actually a little bit more optimistic. Foundation Medicine, who are a diagnostics company purchased by Roche in 2018, they still operate independently as Foundation Medicine, but as part of Roche. And then Okairos, which were based in Switzerland, were a vaccine company purchased by GSK in 2013. Their technology is still out there. I think GSK out-licensed a bunch they were looking at things like Sudan Ebola and various other things. It was out-licensed to an institute, and I think work is still going forward on that vaccine technology. So although you have these like categorizations, it's still a little bit gray.
Yeah, well, and I think you're right, but I think so if there's really just maybe 4 or 5 where the therapeutic is just no longer being used, there's 4 or 5 of them that are still in use but under a different name.
Yeah.
And the remaining 5 are still working, right? And
So
the biggest name on here, right, it took us long enough to get there. It's Bluebird, which
absolutely.
They had the 2 approvals, so that ties in nicely, and that's a great story, right? Like hats off to Fierce to identify them back then. And
so Bluebird, basically that you're right. Bluebird are the probably the success story of this,
and Mersana I think is right up there too, but that's okay. Maybe I just like them. But no, Bluebird is definitely. Look, Bluebird is the largest of them on here, undoubtedly. Yes, largest value, I would imagine. Their cash value is the highest.
So the ones that are still going, you got AC Immune, where they basically. I mean, AC Immune, part of the issue is they've chosen a really tough disease.
They did.
They've got Alzheimer's.
Yeah.
They're mainly focused on monoclonal antibodies, but I mean, if you look at the website, they've got a ton of stuff in the pipeline. They're still going. They're partnering with Genentech, Lilly, and J&J, and they actually, as part of this, I was just cleaning up yesterday. They announced preliminary data on an Alzheimer's vaccine.
I think their stock shot up. I feel like something happened. One of the, I think it's them. But you're right. They're in the tough. It's impressive they're still around relative to that space.
So then you got they're still going. Alkeus Pharmaceuticals in Boston, looking at ophthalmology but genetic diseases.
Mm.
Their lead molecule's still in development, so they're
Another
still
tough
going.
area too, right? Like, oh, great.
Then Bluebird is the two approvals, and
Mhm.
obviously active in the gene therapy space. Enanta
Yeah, we're
are still going.
strong company, right?
They were an infection company that who again, you read the story, had a lot. I think we're doing well, but they went after a difficult target. NASH. You look
yeah.
at the number of companies who've crashed and burned in the NASH
Well,
field.
let's talk about that for a moment. Both Alzheimer's and NASH, right?
Yep,
Non-alcoholic syndrome
it's it's,
for
it's like
hepatic. Yeah, it's
yep,
basically non-alcohol induced liver damage, which is usually
yes,
due to
yes,
weight,
yes,
right? It's a weight
yep,
thing. But those diseases are more disorders, right? Their origins and the way they manifest themselves are different from patient to patient. So that's why they're so hard to treat, right? Alzheimer's is
yeah,
that like
yeah,
snowflake,
and
as well as
Alzheimer's
that.
was Alzheimer's. I think it was who was it? Someone was talking. Oh God, um, someone was talking, saying, "Oh yeah, the CEO of Roivant was talking
Oh yeah.
recently, saying that they have not touched Alzheimer's because it's still unclear what's
Yeah.
the mechanism, what's behind Alzheimer's, what's the mechanisms driving it. So they
It's
don't. They had like one foray into Alzheimer's that
and
crashed and burned,
the number of
and
biomark yeah the number of biomarkers that come out of this space is unreal, and it's too hard to predict at this stage. Yeah.
So, yeah, but anyway, Ananta have candidates in the pipeline, a couple of which are one for COVID. Again,
Mhm.
touching on what we were saying earlier, that there's still a lot of focus with companies all over the place looking at COVID, and then RSV, which again has been a
Mhm.
lot used
Mhm.
recently with its spike up in the respiratory infection season. Then let's see. I would say Foundation still got Mersana, like you mentioned, with the ADCs, they're still going.
Very good.
Active
Yeah.
clinical studies, again, a lot of partnerships with GSK, J&J, Merck KGaA again, and Asana Biosciences. Meridian are now Viridian, and
Yeah.
yeah, I think
Maybe a quick recap. Oh, go ahead. You said.
those are the ones that are. I mean, of the 15, you got 5 of them are still actively
Yeah.
independent, but there's got others that are active as
Yeah,
part of
about 10 of
larger.
them are active in some capacity. 5 are still independent, and then you got 5 that are truly closed. It looks
Yeah, sure. Yeah,
and
yeah.
then the breakdown, the small molecule, man, it still dominates, right? One, two, three,
Well,
four,
I
five,
mean,
6, and then 7. Technically 7. That peptides, the angiochem falls into
yeah.
that. So 7
10,
small molecules, right?
10 years ago, that's not surprising.
No, I know
Yeah.
it isn't, but there's two, technically 2 of the ADC on there, maybe 3 protein based, but
Hmm.
2. And then Johnny catch there's 2 gene therapies and 1 vaccine. That seems like almost like today's trend, right? It hasn't.
Sure. Yeah.
It's shifted
Yeah.
a little bit more in gene and cell therapy, and small molecules dropped off a fraction, right? But that surprises me. Not, I don't know if it surprises me. I think this misnomer around small molecules being dead, we should just stop talking about it, because what's happening is the chemists are digging in and they're finding bigger, better ways to make the small molecules. So good for them.
And you got to look at this though. This is again, these are the, I mean, these are the things that have legs, and it's probably like some really new and cutting edge. I think it's less small molecule is cutting edge, but there's still a need for them. And I think the more relevant numbers is how many small molecules are approved on a year-to-year basis
Yeah.
as a proportion of the full approvals.
Well,
To
John,
tell you how small molecules are.
yeah, sorry to talk over you there. What a fun podcast. This one I really enjoyed. We're winding down, right? We're right at the hour here. Um, but I want to switch gears. I don't know if you had any closing parts, but
I
I
think
really enjoyed that with you.
one last thing I'd like to say is I threw it that we have the topic of KCS's potential partnering. The fact of the matter is that for all these categories, as the industry's developed, we've developed, and we can really provide bioanalytical support to every one of these companies, regardless of their platform, because we've got expertise in place to handle that. There's my salesy bit.
No, I think your sales event. It's our vision. It's what you've been here, what, 3 years, I think. I've been here almost 10, right? The whole part of what we're doing is we want to be able to say we can cover any PK or PD or any sort of quantification of a drug independent of its modality. And John, we are ever closer, right? We got high res mass specs now. We got PCR, digital drop PCR, and then next gen sequencing. And then I think we'd be able to say we have everything. I think we'd be hard pressed to not be able to. But after we get that, not only that, stay tuned, John. I think we have some access to next gen sequencing that we're working on, so we'll be able to do that. But I love it, John. It's not salesy at all. I think it's important to mention
because as a bioanalytical person, that's what excites me. Is like I can go into a conversation with a potential customer, and they can say, "Here's our problem," and of course I want I'd love to do it by LCMS, but it's really, I mean, if we can, it's also nice to know that yes, we can handle that by another approach, because we can truly help you as a full service full service on the bioanalytical base.
Yeah, it's what differentiates us from our competition, John. That is exactly what differentiates us. We have expertise in just about every platform for every drug. And even though we have sites across the world now, one of the hallmarks is our headquarters now in Olathe, Kansas here, where it's 70,000 square feet. It's the fourth largest bioanalytical laboratory in the continental U.S. That's a proud statement for us. And it's all under one roof. So all of our services can be done under that roof. And so, it's wonderful. It's a very proud moment. So John, I'm super excited to talk about the weekend, and I hope you know why I'm excited to talk about the weekend. I don't know if you've heard it yet, but the Kansas City's hosting a AFC championship game. Did you know that?
I sort of knew it offhand, but I really haven't paid any attention.
Well, I want to take a step back, because last Saturday, guess who was at the Chiefs game against Jacksonville for the divisional playoffs round? Well, this guy. I went with I took my son and one of his friends, and one of my neighbors went, and actually two of my neighbors went, and then a group of like 5 or 6 other friends. So there were about 25 of us at the game, and we had a good time together. And it was weather was decent. More importantly, the Chiefs won by a touchdown, and Jacksonville ended up covering. No, I'm just kidding. And so, this weekend is the big game. I'm not going this weekend, but I'm sure I'll find my way to some AFC championship parties or something, but that's got the city a buzz here.
I'm sure
we are so fortunate that our team, this is the 5th year in a row that Kansas City's been in the AFC championship game. I did go to one a couple years ago when the Patriots won, but that's consuming the weekend. You can tell me a little bit more, but then I want to tell you a little bit more about some of the awesome food I've been making.
Well, Dom, this is Jeremy. I want to break in for a second, because I don't know if I told you or not, but I am actually going to that game this weekend in Kansas City.
You did. You went last year too, right?
Yeah, that's right. Because my wife she's a Cincinnatian, and she's a big Bengals fan. So when we heard last year that the Bengals were playing the Chiefs in KC, we went, and when they won, actually everybody from Cincinnati was pretty cool and easy to get along with. But
yeah, they flooded the city last year, Jeremy. I rember distinctly being out for brunch on Saturday, thinking, "What the hell are all these Bengals fans doing?"
I know, right? And so this year, when we heard it was going to be the exact same situation again, I told a couple of my buddies that I was coming in, and we're hoping that the Chiefs can redeem themselves from last year. And they said, "Well, listen, Jeremy, if they lose again, you're not allowed to go to another playoffs game in Kansas City."
Well, Pat Mahomes appears to be healthy, but that's the biggest question right there.
Yeah, sure. I did see the part of the game against Jacksonville. I was because we went out for food, and the bar was playing the game while we were eating. So
yeah, my son's a Jacksonville fan, so he actually dressed up in Jaguar gear, which is funny.
Yeah, my wife did the whole Bengals thing too, which is it
Be
made it
safe,
a little rough
Jeremy.
walking around.
Be safe. I know I kept him a little bit closer, and that was a good moment where I'm happy I'm a 300-pound man. Not
Yeah.
often, but that day I was. So
Yeah.
so John, I'm gonna throw it back at you because I see the FA Cup's coming out in the Premier League this weekend, and guess who's in the fourth round? I don't know if you saw the team that's owned by Ryan Reynolds and the other
Oh,
actor there,
Wrexham. Yeah,
Wrexham.
yeah, yeah,
Yeah,
yeah,
they
yeah,
made.
yeah,
I
yeah.
can't believe it that they believe everybody laughed at them when they said they're going to be in the Premier League. Well, they're not in the Premier League, but I got to tell you, making it in the fourth round of the FA Cup,
Oh,
that's something.
the FA Cup is great because
It is. It
it
is.
throws up these upsets that are just can
Yeah,
be
I
incredible. I
noticed
mean,
Chelsea wasn't in there. They're Chelsea's
yeah.
garbage.
Well, I mean, it's like Hereford
Yeah.
fans dine out on their FA Cup moment from 1972, and we still do it. And it's like it's it's one of the best, one of the big. Well, I think it's possibly. Oh no, I think it's the biggest giant killing ever in the
Yeah,
English
yeah, I
FA
think it
Cup.
does. Yeah, that
But
game in the slop, right? It
yeah,
almost looks like it's black and white, and they're in the mud. That is
yeah,
the
yeah.
biggest upset.
So it's the biggest giant killing ever. Plus, it had the goal that is played every year on on TV. But in terms of giant killings, obviously you got the FA Cup going on in England. I don't know if I probably wouldn't have come across, but there was a couple of Scottish Cup games because obviously Scotland has their own competitions in parallel with England, and there's a team called Darvel.
Hmm.
Or Darvel. I don't even know how to pronounce it because I've never heard of the town. Beat Aberdeen, and there's 6 divisions below Aberdeen, and beat them 1-0 in the Scottish Cup, which is being billed as the biggest giant killing in Scottish football in 150 years. So it's like, yeah, that happened just this week. So
Love
I
it.
mean, it's these cup competitions can be amazing, when they throw up these unpredictable games and
They wouldn't even do
conditions.
that here in the U.S. It's too risky.
Yeah,
I
conditions.
mean, it's too risky for the pro teams to
Yeah,
play that level.
conditions. Yeah, it's like conditions may or may not work against the
Uh-huh.
professional team. And I think Aberdeen were going through a slump, and it was like timing was perfect for Darvel. I will have to find out how it's pronounced. And yeah, they won.
That's great.
It's incredible.
Well, so
So yeah,
that's my excitement, and then I'm going to share a little bit because I can't not talk about pizza. I don't know what's going on with me, but my wife and son, I made actually, I'm now able to like make a pizza pretty quickly, and they told me I made my best pizza
oh
ever
good.
on
Yeah.
Wednesday. I actually used the oven stone and not my uni or anything. I make they say my pasta sauce, and then I've been mixing cheeses, so I make my own pasta sauce. I've been buying the dough from a place called Waldo Pizza, and so I freeze it, and so I've got this awesome dough, my homemade pasta sauce. I don't use any fancy cheeses, but I mix them a certain way, so it's still just like craft cheeses, but I make this little blend that I like, and then I buy good pepperoni, and somehow, they were like, "This is the best pizza you've ever had," and I was like, "Wow." That made me feel good
So
this week.
So I've been on cooking fire lately too, and later in the year I'll maybe give the reason behind it. But so in the last, so one thing I've always done is make Scotch eggs, which are basically an egg surrounded in sausage meat, breading, and then deep fried. It's an art to cook boiling. I do it with quail's eggs,
Mm.
so you basically boil, peel, wrap in sausage meat. And I've tried various blend. I did a bunch before Christmas. Didn't like the sausage blends, so I decided I'm going to start playing around with sausage blends. And did two the same day. One was like a spicier based on a Cumberland sausage. It was like much more nutmeg, mace, and things in there. And then I did another one based on Lincolnshire sausages. And the recipe I tend to work from recipes and then tweak them where I see fit. And this called for pork and bacon fat, not back fat. Didn't have back fat, so I used some like a blend of British and US bacon. Those were fantastic.
Mm.
I was like, so we just finished the last one, and we're gutted about that. But then I moved on to in the last week I made a smoked salmon and spinach lasagna, which was
Oh
great.
wow.
A chicken and broccoli bake, which wasn't great. A beef and mushroom and beer stew with like mustard mashed potatoes, which was great. A cheese and leek flan, which is a Welsh recipe, that turned out really well. Last night I did a chicken tikka masala, and right now I have the batter merging flavors in the fridge to make cannellis, which is a Bordeaux.
Oh, wow.
Bordeaux.
You're a little more sophisticated. I've been making chicken ziti broccoli, if you know what that is at all. That's but I've been sous videing the chicken, right? So then chicken ziti broccoli, chicken palm. That's another one, a hallmark of mine. I've been branching out a little bit. I made a lobster mac and cheese. We had that the other night, and I have been certainly trying to eat more salmon, but it's usually just salmon over a bed of lettuce or something. But you've got some really eclectic food in there. I will let you know that.
What I like to do is I have a bunch of recipe books, mostly from like chefs, etc. And I like to go at their recipe first time. And if I see ways I want to tweak it, then I know where I'm going to go.
Well,
even with the chicken and broccoli bake that we did, my wife and I are eating it because like there's something not right here, there's something not right here. But while we're saying there's something not right, we're like, but you could do this, you could do this, do this. So we're already going to do it again.
Yeah.
It's just
part of the fun, right? That's part of the fun is trying something new and it not being so good, but refining it. There's the chicken ziti. I actually don't use ziti, but chicken broccoli and just pasta. I've made that a few times in my son's 17, so it's not hard. I get more, and if he likes something, I tend to make it more. And then, I'm all into my green egg, so I'm looking at my green egg book. I make wings all the time too, and smoked some ribs recently. But then when we went to the football game, I did bring. I imported meat from Nebraska because right, but they have these hot dogs. They're called some people call them pop-up dogs, but they're made by Weimer's and they're red casing, and you can't buy them in Kansas City. So when I pulled them out, a lot of Nebraskans around here, they're like, "Oh my God, where'd you get the red dogs?" I'm like, literally, I muled home about 20 pounds, sold 10 of them to a friend because he wanted them. And then you mentioned sausage. And there's a butcher shop right near where my wife grew up back in Columbus, Nebraska there. And I bought home about 8 pounds of breakfast sausage patties because they're just delicious. They're high quality. And so my breakfast every morning, and I've been trying to intermittently fast, and I've lost a lot. I'm losing weight. I hope to be under that 300 mark, but and I should be there soon, but it's a couple sausage patties and 2 fried eggs, and that's my breakfast. And then I tend not to eat again until dinner around 7. So I kind of fast during the middle of the day, try to get a workout in late. But anyhow, I'm telling everybody my life story. John, let's wrap up. Looking forward to seeing the Chiefs kick some butt this weekend.
Hopefully do so, yeah, absolutely. Yeah. Yes.
We'll see you in the funny pages.
Good luck to them, yep. And I think for us, it's our big highlight is going to be trying to tie down catering for my daughter's wedding in the next few days, because we the venue said we need to have a caterer lined up at the end of the month. We don't have a caterer lined up.
Well, maybe you could cook, John.
Nah, I'm way too slow. I really love doing it, but I'm way too slow.
So that makes me feel a little better because I got to get that dinner ready by like 6:30 or 7, or my son is losing his stuff. Sometimes I like to eat a little bit later, but like that does condense the meal time prep for me. But anyhow,
I mean, I'm getting better, but I could never work in a restaurant or anything like that. But I am enjoying doing different things and learning different approaches.
Well, I think you could work at a restaurant if you didn't have to make a living doing it.
Yes.
All right, John, take care.
Okie doke. Yep. See ya. Thanks very much.